Good afternoon to all. I am very honored to stay here, and thank you all the organizers to give me the opportunity to show you our results, our study on the role of antigenic molecular mimicry in anticancer immune response. I am not the disclaimer. These are the research activity at our lab, the Innovative Immunological Models Lab in National Cancer Institute in Naples, in Italy. Fundamental, we are focused on the identification of specific tumor antigens for development of innovative therapeutic and preventive immunotherapy, especially on therapeutic cancer vaccine. Our goal is improving antigenicity and immunogenicity of tumor-associated antigens. And we published several studies on the design combinatorial strategy to improve immunotherapy efficacy. For example, in preclinical setting, we combined immunomodulatory drugs, for example, metronomic chemotherapy, radiotherapy, or immunocheckpoint inhibitors, to potentiate the efficacy of cancer vaccine. And so we conduct early stage phase clinical trials. We finished 2 years ago the first, the EPAVAC vaccine in patients in a European consortium in patients with hepatocellular carcinoma. And we obtained promising results, and just in this week we start with the second clinical trials where we combined therapeutic cancer vaccine with immunocheckpoint inhibitors. But to develop a cancer vaccine is important to identify and validate target antigens which should be able to be able to induce a specific potent T-cell response against the tumor cells. And these tumor antigens should be specific for the tumor, so they are expressed only on the tumor cells and not to the normal cells. There are two main groups of tumor antigens, the tumor-specific antigens and the tumor-associated antigens. The tumor-specific antigens give rise from genetic alteration, the mutated proteins in the tumor cells. And there are specifically expressed only from tumor cells and not from normal cells. But their discovery for their recovery is essential to apply a high throughput omics screening analysis. In general, these analyses are very expensive and so not applicable to large scale. On the other hand, the tumor-associated antigens give rise from mutated protein, from overexpressed protein, and so can presented by also from the normal cells. So they generally have a low immunogenicity and are affected by a central and peripheral immunotolerance. For this reason, it is important to improve their immunogenicity and the antigenicity, designing, for example, the analog peptide in which specific amino acid residues are substituted to improve their antigenicity, namely heteroclitic peptide. We demonstrated that this modification could activate a high strong T cell CD8 T cell response. And then found the non-self antigens derived from pathogens, bacteria, and viral, with high sequencing and structural homology with the tumor-associated antigen. So the molecular mimicry, the concept of molecular mimicry. The molecular mimicry is the mechanism by which infection agents may induce autoimmunity. It occurs when similarity between foreign and self-peptide favor an activation of autoreactive T and B cell response. In the past, the concept of molecular mimicry was associated to autoimmune disease, was described from several groups in the past to describe the autoimmune disease induced by the microorganism, and four major criteria that define this concept, the molecular mimicry. So the similarity between a host epitope and epitope of a microorganism, the detection of antibody or T cell that cross-react with both epitope in patients with autoimmune disease, the epidemiological link between the exposure to the microbe and the development of autoimmune disease, and obviously the presence of autoimmunity in animal models. But molecular mimicry in the past was described for the first time by a group of Alicia Livottini, where they, in this work, they described for the first time the sequence homology between a tumor-associated antigens MART1 in melanoma and un herpes simplex virus type 1, where in the central part, in central sequencing of monomer presented by the HLA molecules on tumor cell, had a high homology to the virus, the viral epitope. Here is a crystallographic analysis of structure of CD8 T cell TCR, the epitope, and the groove of HLA 0201 HLA. And we can see that there are specific amino acid residues very important to have the cross-reactivity between by the T cells. So there are these residues interact with directly. Is very important because interact directly with the TCR of CD8 T cell. The TCR cross-reactivity is explained by several experimental data that estimate a single TCR can recognize at least 10 to 6 different MCC bound peptides. Such TCR degenery is necessary because if each TCR could recognize only one target, the number of required single naive T cells carrying a different TCR and so would far exceed the number of cells in the human body. So we believe that pathogens can be our allies against cancer, because if the antiviral memory T cell recognized on MHI, the viral epitope, which can activate a specific antiviral T cell killing infected cells. In the same manner, the anti-tumor T cell recognized a specific tumor peptide and so can kill the cancer cells. And a cross-reactivity antiviral and anti-tumor T cell can kill the cancer cell. So a pre-existing memory viral T cell can recognize the same and cross-react with a epitope that shared a high homology with the viral peptide. So we start in discover the homology, the study in screening the homology of tumor-associated antigens and viral epitope. And in a very short cohort of HCC patients, we found a high homology between the tumor-associated antigens, the neoantigens. In this cohort, we studying the neoantigens and several viruses. In particular, in these patients, we found that 3 neoantigens had a very high similarity, especially in the residues that which interact with TCR of CD8 T cell, with vaccinavirus, with Shigella, and with the Lassa virus. And we performed to validate this similarity and the effect of this homology. We validate ex vivo the T cell specific reaction by ELISpot analysis, and we found that the PBNC of these patients are able, after stimulation in vitro ex vivo with vaccine with viral epitope, can recognize and can activate the T cell specific against the mutant the neoantigens. Then successively we identify and validate all tumor-associated antigens from the cancer antigen peptide database, and we focalize our attention on 99 tumor-associated antigens. We found 82 homologous viral epitope in particular, the major represented by HIV and herpes virus, and only 20 of these viral epitopes are a strong binder. So they are potential antigen immunogenic peptide. In particular, you can see that, for example, for GP100 and tumor-associated antigens in melanoma has a high homology in sequencing, but also in structure with human cytomegalovirus epitope. You can see here, for example, the binding residues are to MHC and to TCR, the yellow and the red residues corresponding to beta and alpha chain of TCR are completely the same of the viral epitope. So, and also, for example, the CHEA antigens are similar, are very homologous to two epitope of M protein of HA1. And then we validate this prediction data by ELISpot analysis, and we observed the cross-reactivity of T-cell response against the tumor antigens and viral antigens. Then we focalized our attention on a cohort on tumor-associated antigens found, described in epitheliocellular carcinoma, and starting from about 5,000 LARF protein, non-detected, present only in liver tumor and not in normal cell, we arrived to detect 9 protein with high expression and correlated with unfavorable prognosis, and focalize our attention on these 9 protein. We found that the epitope of give rise from these 9 protein are similar, are homology to HCV, for example, epitope, and to HBV and other viral antigens. We perform also in this case the structure and conformational analysis to demonstrate the same structure and the same residues interact with the TCR. We perform in using a TAP deficiency 2 cell the in vitro analysis of binding affinity and stability. We use an antibody against HLA 0201 expressed on these cells, and we observed the high stability of both tumor-associated antigens and corresponding viral antigens. Then, to demonstrate the cross-reactivity between the T cell, the CD8 T cell activated from tumor-associated antigens and viral antigens were the same. We used the Immudex dextramer technology. We loaded the dextramer with the tumor-associated antigens and with the corresponding viral antigens, and performed the flow cytometry analysis with dextramer. This is an example of the gating strategy that we used, and how you can see in this example, and for these tumor-associated antigens and corresponding to the viral antigen, we observed the higher response to tumor-associated antigens and to viral antigens, but a high cross-reactivity of this T cell. Because we found a lot of homology between tumor-associated antigens and HIV peptide, we focalized our attention on this on HIV sequencing, and in particular starting from the assumptions that several study lecture to report that the patients with the longest survival patients with HIV and AIDS have a low incidence of these three cancers, breast, colon, and prostate. We analyzed the homology of the tumor-associated antigens in these cancers with the HIV, and we found several homology. In particular, we found a high number of strong binders of HIV peptides in colon and breast, and for example, also in colon we found the broad specificity of these antigens with broad HLA types. And if we focalize the attention on the different tumor-associated antigens, we found, for example, the broad number of strong binder of HIV peptides in CCR9, which is a tumor-associated antigens of breast. Also, we validate our prediction analysis with experimental binding of tumor-associated antigens and HIV paired antigens. We confirmed the same data with conformational and docking analysis. And we demonstrated ex vivo the response and the cross-reactivity of T cell activated from tumor-associated antigens. In particular, we perform vaccination in vitro. We stimulated the PBNC of negative of healthy donors and HIV positive patients with for 10 days with HIV peptides, and then we loaded the tetramer with the tumor-associated antigens, and we observed that in HIV positive, we observed great background also in untreated PBC, because obviously there is a memory T cell against the HIV peptides. But we observed when we stimulated with the treated the PBNC with tetramel loaded with the tumor-associated antigens, EpCAM or CCR9, we observed in HIV positive patients a high response rate. We then obviously analyzed also the homology of tumor-associated antigens with microbiota antigens, and we found interesting the homology with the Firmicutes and the Bacteroidetes classes of microbiota. Which represented 90% of all microbiota of our body. We confirm also for these antigens the cross-reactivity, as shown here, of both of T cell response activated by the viral antigens, the microbiota antigens. And finally, I want to show you a very preliminary data with the antigens from SARS-CoV-2. We are analyzing the PBNC of positive affected by patients affected by SARS-CoV-2 and vaccinated people, and we found also a high homology with some principal tumor-associated antigens and the 5 protein of SARS-CoV-2, the ORF1 and the glycoprotein membrane of SARS-CoV-2. The next step to validate our result and is obviously to sequence this TCR by single cell analysis. And in conclusion, I can see that the sequence and the structural homology between tumor-associated antigens and viral and microbiota antigen is found in a significant number of cases. T-cell elicited by viral and microbiota antigens are able to cross-react with homologous tumor-associated antigens, and this is very important to open new horizon in cancer immunology and immunotherapy. The natural infection, so we can say that natural infection may represent an anti-cancer preventive immunization, and the net result would be the elimination of a cancer cell in the very early stage of tumor progression, preventing the tumor from progressing. The microorganism antigens with homology to tumor-associated antigen represented a novel class of highly immunogenic universal antigens to be used in cancer immunotherapy. So this is my group in Naples, and thank you for the attention.